So in this slide, we are going to highlight the sodium glucose transport system in the context of a 3-transport overall system that allows us to move glucose from our intestinal lumen, from our gut tube, from the food that we've eaten at a meal, ultimately into our bloodstream. And it's really important that we get as much nutrients as possible out of our gut tube into our bloodstream. So how do we do that? How do we make sure that we do and move all of that? So to highlight this, we are going to discuss 3 transporter systems that are going to be a great way to review the kinds of transport that we have, whether it's uniport, symport, or antiports, that we're going to talk about facilitated diffusion versus primary and then secondary active transport. And then ultimately, we're going to put this together in a story to explain why we do certain things when we are talking. We had this with our cholera toxin case study. Why we have to do certain types of things to be able to rehydrate somebody, why Diet Gatorade is really sort of a fallacy and a farce. Okay, so the sodium glucose transport system is this guy that we have right here. I'm going to label it number 2 because it's actually going to be the second guy that we talk about, but highlighting, it's abbreviated SGLT, sodium GL glucose transporter. So that's why it's called SGLT. But we need to couple it with a sodium potassium ATPase. Okay, that's going to be a primary active transporter. And then we're going to have a glucose uniport that we have here. So we're going to put all 3 of these together in a story to understand what's happening. So before we do that, this is a good opportunity to kind of review what we know about your gastrointestinal tract, your GI tract. Your small intestine is a really important portion of your GI tract that allows for the absorption of nutrients. Remember, your GI tract is really just a single cell thick, and we're going to see that here. So here you have kind of your small intestine, this lumen space, that's the inside space. Remember, that gut lumen really is the outside environment, because from the start of your GI tract all the way down to the end, it's just a tube that is the outside environment connected to the outside environment, okay? But when we sort of zoom in here, as we see that we have these folds within your small intestine that kind of create these larger surface areas here. And then we have these villi here, which these are where we have these single cells that we have here. That's the single cell of your GI tract. And so these are what we call brush border cells. Okay, and they have these little microvilli that we have here. But importantly, we talked about the importance of the integrity of this tight junction between your intestinal epithelial cells. Okay, so that's a really important piece to kind of maintaining your gut integrity and not having what we called leaky gut. Okay, and it's important then that we have transporters that are going to direct and control what's getting across this important barrier. Okay, so we're going to talk first about this sodium potassium ATPase, and I'm just going to walk through it here, and then we'll put some words down to sort of put this together. This sodium potassium ATPase is important because it builds up a sodium ion concentration gradient. So it is going to be pumping sodium out, so we're going to have a low concentration of sodium inside. Why that's important is a higher concentration of sodium outside is then going to be able to come in, and we're going to use that dissipation of that gradient as a way to piggyback and bring in glucose on our sodium glucose transporter. And then the third step is, well, once we've packed in high concentrations of glucose inside your intestinal cells, then we're going to be able to export that to the bloodstream, and then we're going to be able to get that glucose that was from your food into your bloodstream and ultimately to your cells. Okay, so let's talk about these transporters one at a time and make sure, highlight what kind of transporter is it, what kinds of transportation is it doing, and then what's the net effect of what's happening. So this first transporter here, the sodium potassium ATPase, is an antiport. It is moving 2 things in opposite directions. And because it's moving each of those against its concentration gradient, it's active transport. And because it's using ATP for that process and moving charged species, cationic species, it's primary active transport. So this is a primary active transporter. And this primary piece, okay, this primary piece is indicating that the cost for this, the money that we're using for this, is going to be ATP. And we are moving charged species, specifically cations. Okay, so the cost for moving things against their concentration gradient is going to be paid for by ATP. So here's the net of what we are moving then. We are going to have, so that we are going to have low concentration of sodium inside our brush border cell. So this is sort of the net effect of what's happening. And we are going to have high concentrations of potassium inside our cell. So sodium is being pumped out, potassium is being pumped in, and the net effect is we're going to have high concentrations of sodium inside, low concentration of sodium inside and high concentrations of potassium inside. Okay, and that is because we are taking and pumping sodium out. So sodium is pumped out and the potassium is pumped in. Okay, so we need ATP for this process, so we need some money, biological money for it. We're pumping sodium out, potassium in, and we are building up both of those concentration gradients. Okay, so let's move on now to our SGLT transporter. So that SGLT transporter is a symport. We are moving two things in the same direction, so it's a symport. We also can think of symports as sort of piggybacking. Okay, so this symport is a secondary active transporter. And secondary active transport differs from primary active transport is the cost of this is another ions gradient. So specifically, it's the gradient that we just built up with sodium. So secondary active transport, the money is another ions gradient, and in this case, it's going to be our sodium gradient. Okay, so the money that we have here to pay the cost is that sodium gradient that we just built up. And so secondary active transport is going to be moving uncharged species. And then specifically, it is glucose in this case. Okay, so I want you to think about what we really have here is we do have a higher concentration of glucose in that lumen space to start, but we want to make sure to pull in as much of that glucose as we can. So the goal is to have a much higher concentration of glucose inside the cell than we have in our intestinal lumen. So to start, we might have glucose concentration in our lumen being higher. I can't write arrows right now. Higher, okay? But we want to make sure that we push that glucose all the way in. So the goal is that we want to have a very high concentration of glucose in our brush border cell. Okay, so that's the goal of that secondary transport is we want to have a very high concentration of glucose inside our brush border cell. We don't want to leave glucose in our intestinal lumen. That would be a waste of the food that we ate. All right, moving on to the third piece now. Now we're going to have that glucose uniport. So it's moving just one thing, and it is facilitated diffusion. So it's moving glucose from high concentration to low. So again, this is facilitated transport. Okay, and basically we are going to use that high concentration of glucose that's in the cell, and we're going to move it out into the bloodstream. So we're going to use that high concentration of glucose that's in the cell. That was what we just built up, that high concentration of glucose that's in the cell, and we're going to basically put that out to glucose in our bloodstream. And so that is the goal. We started with glucose that was outside in our intestinal lumen. Should show it here, glucose that's outside in our intestinal lumen. The goal is to basically get it out into the bloodstream. So a couple of things to tie together with things that we've already talked about with this transporter. First is it needs sodium with it. So if we want to think about this concept of diet Gatorade, if you will, remember the purpose of Gatorade is an electrolyte replacement solution. So if we want to have sodium that we get back into our bloodstream, we need to take it in from our GI tract from things that we drank, right? So if you've got an electrolyte solution, if there is not glucose that is there, you can't piggyback those in together. So glucose is absolutely essential to get the ions out of your blood or out of your intestinal lumen and into your bloodstream. So diet Gatorade, not really doing what you think you want it to do in terms of getting electrolytes back into your bloodstream. So got to have that glucose with it in order to do so. The second thing to highlight is we did see this SGLT transporter when we talked about our cholera toxin case study. So remember, how cholera toxin works is it binds to a receptor that's on our small intestine epithelial cells, and through a cascade of events, just refreshing.
I found this on the web for as it binds to.
What we really have is after we have that binding, we talked about the cell signaling pathway that we activate adenylate cyclase. Okay, we activate and then produce more cyclic AMP. That cyclic AMP is going to activate pKa, which is our master phosphorylator. Then we're going to ultimately upregulate the activity of that CFTR, that cystic fibrosis translocator protein, and that's going to put high levels of chloride out into the GI tract, and then that's going to have water simultaneously go with it to try to balance that change in ion concentration there. Putting high amounts of water into your GI tract are what cause that diarrhea. Okay, so that was that CFTR transporter. We'll have a short video to kind of refresh our memory on how cholera toxin works. But ultimately, the piece that's important and related here is if you want to rehydrate that individual, and if you're going to be doing something orally, you have to make sure that we've got sort of glucose and sodium in there if we want to be pulling those ions in and maintain that electrolyte potential imbalance that was happening, is we have to have glucose there to be able to sufficiently help with that rehydration and that electrolyte imbalance situation. Okay, so that's what we have to wrap up our discussion of our sodium glucose transport system.